Well, we just talked about transcription, so let's go into translation. Translation is the production of a new protein from a messenger RNA molecule that was just produced from the DNA. And so we need our messenger RNA, we're going to need our ribosomes, and we're going to need those transfer RNA molecules. So the messenger RNA leaves the nucleus, enters the cytoplasm, and immediately embeds or threads itself into a ribosomal RNA, or the ribosome. The transfer RNAs now are going to move to a nuclear or an amino acid pool, so different amino acids, and they'll pick up the proper amino acid that they code for. And then they bring the amino acid to the ribosome for the production of proteins. So when we read our DNA, we read it 3 nucleotides at a time, and that codes for that produces a codon, and a codon is what codes for our amino acids or our the protein, the reading portions of our protein. This is a codon table, and it's written in messenger RNA, or I shouldn't say in messenger RNA. It's written in RNA. So instead of having T's, we have U's here, okay? When you're reading a codon table, because we're just reading 3 nucleotides at a time, we have the first letter in this first column. So if the first letter is a U, you would look in this area. If the first letter is a C, you'd look in this area. If the first letter was an A, you'd look in this area. And if it was a G, you'd look in this area. The second letter then runs along the top in the first row. And again, you have UCAG. So if the second letter is a U, you'd look in this area. Oh, darn it. If it was a C, you'd look there. G, you'd look there. A, I'm sorry, you'd look there. And G, you'd look here. And then the third letter of your codon is going to be found over here. So we have UCAG, 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 UCAG. So, sorry. All right, so let's just pick 3 nucleotides. AUG. AUG it codes for methionine, and it's always the start codon. So if we know the 3 nucleotides that make up the start codon are AUG, we can look over here in the first letter. We find the A is right here, so we know it's in this row, one of these rows right in this area. And we go up here and we say, okay, we need the U. So now we know that it's one of these 4 nucleotides. So we go over here and we say, okay, we need the G. So it's this one. So AUG is right here, and it codes for methionine. Methionine is always the start codon. We need transfer RNAs. Transfer RNAs carry an amino acid on one side, and they have what we call the anticodon, or the complement to the messenger RNA, that they can bind to the messenger RNA. So if we're coding for the amino acid methionine, and that's AUG, the transfer RNA would have a U, an A, and a C, UAC, because U binds with A binds with U, and C binds with G. And this is a fancy picture of what transfer RNA looks like. I usually make a stick figure RNA, transfer RNA, and I'll show you that on Wednesday if you guys want to go over that, because this can kind of be confusing for some people. So I can show you how I draw my messenger RNA or my transfer RNAs. They always have, like UAC would have some amino acid up here, okay? And UAC would bind to the AUG over here, okay? And then this is the ribosome. So there are 3 phases to translation as well. Initiation, where the ribosome attaches and at that start codon so that the transfer RNA can attach with the methionine. Elongation, as the ribosome moves along the messenger RNA, more transfer RNAs come in carrying their amino acids. And then as the ribosome moves, so amino acids stay in place. So they are all going to get transferred to that first spot. And they bind to the other amino acids, forming peptide bonds. And eventually we'll have a polypeptide. And then once we hit the stop codons, which there are 3 of, that's called termination. When we hit the stop codons, the ribosomal subunits come apart. The protein, the primary form of the protein, the polypeptide, is released. And we can now either retranslate this messenger RNA, or if the messenger RNA is not needed anymore, it will get degraded. But the ribosomes and the transfer RNAs are not going to be used up in this process. So this is kind of showing initiation, where you have methionine binding to the start codon. And then as the ribosome moves, methionine, a new nucleotide comes in. There's a bond forms between the methionine and it looks like glutamate. And then the original amino acids get kicked out. So now the transfer RNA that carried methionine leaves, and the transfer RNA that carries glutamate goes to the first site, or the P site. Now we have another amino acid moving to the second site, or the A site, and it brings along with it its amino acid. A bond will form, and this amino acid, or this transfer RNA, will move to the P site. This one will leave, and another one will move over here. This occurs until we hit that stop code. Once we hit the stop code, then the polypeptide is free, the ribosomal subunits are free, and those transfer RNAs are going to move back to the amino acid pool to pick up new amino acids. And this we're going to watch.
Break it down. The DNA, the DNA, the DNA makes protein. The DNA, the DNA, the DNA makes protein. The DNA, the DNA, the DNA, the DNA, the DNA, the DNA, the DNA makes protein. DNA, the DNA, the DNA, the DNA, the DNA, the DNA, the DNA makes protein.
The nucleus dissolves when it's time to divide. Nutrigenious bases line up side by side. Sugar phosphate backbone goes along for the ride. String them all together, make a nucleotide. A pairs with T and G pairs with C. It works cause the code's complementary. It lets you be you and me be me. From Coach Jim Tressel to President Gee.
The DNA, the DNA, the DNA makes protein. The DNA, the DNA, the DNA makes protein. The DNA, the DNA, the DNA, the DNA, the DNA, the DNA, the DNA makes protein. DNA, the DNA, the DNA, the DNA, the DNA, the DNA, the DNA makes protein.
Transcription takes the bases that are found in one gene, converts them into RNA, if you know what I mean. The bases pair up just like they did before, but U subs for T, which isn't needed anymore. RNA leaves the nucleus, but the job isn't done. Ribosomes roll in to join in all the fun. 3 bases make a codon, count them 1, 2, 3. An amino acid for each codon in the growing protein.
The DNA, the DNA, the DNA makes protein. The DNA, the DNA, the DNA makes protein. The DNA, the DNA, the
never-ending. Polypeptides ever-ending. And say for random chance mutation, precisely copies each creation. DNA is the star of this fanciful tale, in the smallest mouse, in the largest whale, determining your features into the last detail. From the cellular obsession, life on Earth shall prevail.
The DNA, the DNA, the DNA, makes proteins. The DNA, the DNA, the DNA, makes protein. The DNA, 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 makes protein. DNA, the DNA, the DNA, the DNA, the DNA, the DNA, the DNA, makes protein.
All right. I hope you enjoyed that. I thought it was awesome, but I like that type of stuff. All right. So the last thing we're going to talk about in this chapter is cell division. So cell division occurs when one cell divides, producing two daughter cells. This is really important so that we can replace new organisms, produce new organisms, as well as for tissue growth, so growth in size, and replacement of cells that have been damaged. So some of our cells get damaged very easily. Think of our skin. Our skin is constantly getting damaged. So the cells at the most superficial region are constantly sloughing off of our skin. There are two types of cell division that we talk about. We focus on mitosis this semester and meiosis next semester. So mitosis is division of the somatic cells or body cells, and it produces daughter cells that are identical to the original cells. Meiosis is cell division in our sex cells, so either in spermatogonia or oogonia. And the job there is to produce non-identical haploid daughter cells. Haploid means half the number of chromosomes. So when a chromosome gets ready to divide, when our cells are getting ready for division, the chromosome has to condense. So this is chromosome or the chromatin prior to condensing. And then there's a chromosome that's already condensed, okay? And dude, mitosis starts in 5 minutes. Can't believe you're not condensed yet. Getting all upset, and this chromatin has to get condensed, right? Once the chromosomes are all condensed, they're chromosomes, you have, in the human, we have 23 pairs of chromosomes. So one chromosome from mom and one chromosome from dad of every single type, which makes each of us unique from each other. The chromosomes are going to, or the chromosomes will then go through mitosis, and they move associated with centrosomes. Centrosomes are organelles formed or composed of centrioles, and centrioles are what form the spindle fibers, and the spindle fibers attach to the DNA at a region on the DNA called a centromere. The cell cycle then is the steps that occur in the process of dividing the cell from a parent cell to the daughter cells. So one cell cycle starts with one parent cell, goes all the way through the process of what we call interphase, and then mitosis, and we end up with two daughter cells. And then each of them starts their own cell cycle. So interphase is the first step of the cell cycle. And interphase has three basic periods or parts to it. The first phase is called G1 or gap 1. And when I think of gap 1, I like to use the word growth, because really what's happening is we're growing the cell. The cell is going to get larger. We're producing proteins that will be used in the next phase called the S phase or synthesis phase. Organelles are going to start to double. And then we go through the S phase, which is synthesis of DNA. So S phase occurs and we replicate our DNA. And then we go through gap 2. Gap 2 is another growth phase, so I always think of it as growth, where we're producing proteins. We're checking our DNA, making sure it looks good. We're producing proteins for mitosis. So gap 1 is where we grow the cell's size. We produce new organelles. We produce proteins for replication. Synthesis, we're going to replicate our DNA so that we have 2 identical copies of every single chromosome. And the identical copies attach to their sister. And when they're attached, the chromosome itself is called a sister chromatid. Or it consists of 2 sister chromatids. That's what I should say. They attach at the region called the centromere. And then DNA, let me say this first. DNA replication is how we produce the new sister chromatids, okay? So replication in a nutshell, DNA unwinds, unzips, DNA polymerase binds to the DNA, and we produce a complement to the parent strands. So this is going to happen on both of our DNA molecules. So it's unwound. So we have 2 single-stranded DNA molecules. Our DNA polymerase binds and produces a complement to each side, which then, in a nutshell, produces 2 double-stranded DNA molecules. So here you can see DNA polymerase bound, and it's running along, allowing the proper nucleotides to bind. I don't want to go to a song yet. And then here, this one. Sorry. This one is also bound, and it's moving along on this side. So we're producing nucleotides for both sides, and this is going to make what we call identical DNA strands that are both produced through semi-conservative replication, because they're going to have one parent strand and then they're going to have a daughter strand that is new. The parent strand is old, the daughter strand is new, but they'll be identical to each other. We then enter the second gap phase, where we're going to check to make sure our DNA replicated properly so we don't have any mutations. We're going to produce proteins for cell division to occur, and we're going to then go through the cell or the mitotic stage. Mitosis then has its own 4 phases: prophase, metaphase, anaphase, and telophase. So mitosis is division of the nucleus, and cytokinesis is division of the cytoplasm. Prophase is the first phase, and so in prophase, the nucleus itself starts to dissolve, and the chromosomes condense. So chromosomes consisting of sister chromatids condense. They're held together at what we call the centromere, and microtubules from the centrioles start forming. And they grow out of the centrioles, and they kind of basically start moving towards those centromeres and attach. At metaphase, the centrioles have moved to the opposite ends of the cells. The microtubules are now attached to each of the centromeres. And so now the spindle fibers can move the DNA wherever they want. And so what they do is they move the DNA to this imaginary line called the metaphyseal plate. Each of the chromosomes consisting of sister chromatids lines up one on top of another. And then we move to anaphase. Anaphase is where the sister chromatids get ripped apart and pulled to the opposite ends of the cell. And then we go through cytokinesis and telophase. So telophase occurs when the new the sister chromatids from each of the chromosomes starts to uncondense, forming chromatin again, and newly formed nuclear envelope surrounds each of the new nuclei for each side. And then at the same time, the cytokinesis is occurring where we have this protein that's basically squeezing the cell. The protein is known as actin, and it basically squeezes the cell until we produce two separate cells. So that squeezing is known as a cleavage furrow that's formed. As soon as the cleavage furrow separates the two cells, then we start a new interphase. And so this is the entire process, starting with growth 1, S, growth 2, then we have prophase, metaphase, anaphase, telophase, and we start, and so between anaphase and telophase is when cytokinesis occurs. It's not really its own thing. It occurs here. They're just putting it separate here. And then we start with a new interphase for each of those cells. So this is a really cool song. I'm not going to play it here, and the reason I'm not playing it is because if I do, I think that this person probably makes money off his songs, and they would take my video down, and I'm not going to do that. I don't want to worry about that. So I hope you enjoyed Chapter 4. It is relatively long, but you'll be happy that Chapters 5 and Chapter 6 are relatively short. So I'll see you next time. Have a great day or evening. Bye.